Yeah, now it's recording. Okay, great. So welcome everyone to the online course, Imaging an Ocean of Change. It's organized by ITAPINA, which is an initiative belonging to the Marinets project and co-led by Rainer Kiko, Margot Noyona, and myself. I'm Rubens Lopes, faculty from the Oceanographic Institute at the University of São Paulo in Brazil. I'm speaking from São Paulo right now. I'm a biologist with a PhD in zoology earned in Brazil, with research interests in plankton imaging for quite some time, and a hands-on user of plankton imaging systems since the initial release of the ZooScan as a commercially available instrument that was maybe 15 years ago or more, no more than that. So a long time has gone since then, and it's great to see how much progress we have achieved and how much our community has grown. I think this is something very important. And speaking of that, I'm really impressed by the number of participants to this course. As I mentioned before, to those who have arrived before, we had 211 registered persons from all around the world. And I would like to thank you all for joining us. It's really great you're here. So I hope this lecture series and the subsequent discussions will help you to expand your knowledge on the topic, which is quite extensive and spanning across several disciplines, and also to establish connections and reinforce connections with scientists, engineers, and students working in the field who are also attending the course. So I would like now to ask Rainer Kiko to say a few words about our initiative, ITAPINA, and then I will introduce our today's speaker. So Rainer, can you please?
Yeah, hi everyone. I maybe have a small presentation that I can share. I'll try to do that. Okay, 1 second. Full screen. Around. Oops. So I hope you can see my screen now.
Yes, we can.
Okay, so yeah, welcome everyone to this lecture series that Rubens initiated. As Rubens already said, Rubens, myself, and Margot Noyon are the co-PIs of the ITAPINA Imaging the Atlantic Pelagic Imaging Network Approach project, which is part of the Anchor initiative to build an all-Atlantic ocean community. So my name is Rainer Kiko. I'm Maker of Planet Great-Again Laureate at the Laboratoire d'Oceanographie de la France de Mer. Yeah, and I just want to quickly explain what ITAPINA is about. So ITAPINA is an activity that aims to also support the implementation of the Belém Statement. The Belém Statement was signed in 2017 and aims to further collaborative scientific efforts in the Atlantic Ocean, and signatories intend to sustainably cooperate on marine science, research, and innovation. And this statement was initially signed by the European Union, Brazil and South Africa, and later joined by other countries, for example, Argentina and Cabo Verde. And yeah, so I was at a meeting in Brussels some time ago, and there was a discussion of how actually also in our field of ocean observation, one can support the implementation of the Belém Statement. And so I thought that we could also in pelagic imaging actually do much more to connect people around the Atlantic, because there are many users of different imaging instruments all around the Atlantic, and the digital nature of our science and that we do now enables, of course, sharing of knowledge, of infrastructure, of data, and we work on questions of global significance, so carbon cycle, marine food web, harmful algae blooms, all these issues can also be tackled with pelagic imaging tools. And I mean, also if we share our knowledge, our data, our infrastructure, we actually have a greater reach, and we will have all have access to more data if we collate it. So that's the idea of ITAPINA. It's an open access network for pelagic imaging professionals and stakeholders. And we want to discuss common practices, sharing of instruments, data analysis, data aggregation. And one of the goals that we have in ITAPINA is that we, at some point, that we lay out the roadmap to get to an operational pelagic imaging. Well, what I mean with that is that we actually collect data on ships, on floats, on moorings, etc., and make them available immediately to the community, globally even, so that everyone can work on the data and can really get to scientific output and to advice for politicians very quickly, not with a long delay we have due to the time it takes to get to, for example, a scientific publication. We have now a website where you can find more information about ITAPINA. So I will post this also in the chat repeatedly so that you can find this website at aamari.net and then ITAPINA general info. So if you also just go to aa-mari.net, you will also find ITAPINA there. And you see here as well our goals stated. And you see that here at the bottom there's a button questionnaire. And if you would like to join ITAPINA, you can hit that button and fill in a questionnaire. And we will join, put you on the mailing list of ITAPINA, where you can get information about upcoming events, etc., and also other things, and can of course yourself also post to. And then here we also on the website we have recorded presentations of the first ITAPINA workshop, which actually took place almost exactly a year ago. And also the scientific session that we also had as a aquatic sciences meeting. And there is also a page on online resources where you can, for example, find manuals for instruments, guidelines how to structure data, and for example, also links to different GitHub repositories where data analysis tools and pipelines are developed. Yeah, so I can just invite you to join. As I said, fill in the questionnaire at this website, and then we'll put you. You can join the network, and of course, somehow we actually are open to anyone around the world, because at the end the ocean is linked throughout the world. Or we have a common ocean, and so we will not restrict this to only members that are around the Atlantic Ocean. Okay, with that, thanks a lot for your attention.
Thanks, Rainer. Well, it's interesting to note that among the 211 people who registered for the course, more than half answered that they are not part of the ITAPINA mailing list. So I have included the link to the website where that Rainer just mentioned, and you can have more information there if you would like to take part of the mailing list, and also have a chance to watch the presentations from last year's initiatives. So I realized that Mark Benfield has a long presentation, so we should not take too long to start. But it's really good to have you here, Mark. Thank you so much for accepting to give this initial presentation. Mark Benfield is a professor from the Department of Oceanography and Coastal Sciences at the Louisiana State University in the United States. He has earned a bachelor's degree at the University of Toronto and a PhD from Texas A&M. And for quite some time, Mark has quite active research in pelagic imaging, spanning from plankton to large fish and large invertebrates. It has a very important participation in developing imaging systems and applying them, including his own shadowgraphic imaging system, the ZUVIS, that I'm certain he will talk about. And so his research interests are related to zooplankton ecology in general, optical and acoustic sensing of zooplankton, computer-based classification of zooplankton, acoustical telemetry studies of fish and invertebrates, application of industrial remotely operated vehicles in biological observatories. So Mark, please, can take the floor. Your presentation is already appearing on the screen, so feel free to.
Thanks. Thanks, Rubens. Yeah, this is great to be here. It's great to have this level of participation. It's a little daunting to go first in a course like this because I looked at the list of people who are going to be presenting through this course, and there's some real luminaries there. So what I'm going to try and do today is give you an abbreviated history of plankton imaging systems. When Rubens asked me to give this, I thought, well, this won't be too hard. You know, I gave a review back at a zooplankton production symposium in Hiroshima, and Peter Wiebe and I wrote a review paper on zooplankton sampling and sensing systems, and we did a piece on rapid in oceanography. So how hard could it be? And then I started looking at the time difference, and it's been 15 to 20 years since those things were done, and a lot has happened in that time. So my apologies to any of you who have, or any of the developers of imaging systems that I don't talk about, because there really are a lot of them, and no slight is intended. I just I'm not able to cover everything. So let's get started. So let's see, can you see that? You guys see
I can see your first slide.
You can still see the first slide. Okay, let me see if I can get it. Okay, there we go. All right.
Yeah.
So at the end of the 19th century, Victor Henson, who was a pioneering zooplanktologist, he posed two fundamental questions. What are the numbers and kinds of things in the sea at any given time and place? And how does this material vary from season to season and year to year? And to try and address those questions, he developed the first quantitative plankton net that you can see in that image. So here we are in 2022, and we're still basically attempting to answer these same questions. Now, Henson was armed only with simple plankton nets, but our toolbox has evolved to include single and multi-net systems, plankton pumps, imaging systems, and high-frequency scientific echo sounders. In addition to those questions that Henson asked, we are also asking some additional questions that weren't really possible given the tools he had at the time. For example, what are the scales of patchiness in relation to hydrography? What species occur together and how are they interacting? How are the animals oriented and what behaviors can we infer from these orientations? And of course, I'm sure each of you have your own particular questions that you'd want to address with the sampling and sensing tools. So nets have been a mainstay of zooplankton ecology since the time of Henson. And nets are very useful tools, but they're not by any means perfect. And here we can see a couple of issues with nets. There we go. The first relates to patchiness. Nets are going to integrate everything they retain along their towpath. So we have no idea whether we caught all these copepods in one location or we caught them at a constant frequency throughout the tow. Either hypothesis is possible because all we have at the end of the cast is the number of individuals in the cod end and the volume of water filtered. So in this particular example, we can calculate a mean concentration of about 7 per cubic meter, assuming we sampled 4 cubic meters. But in fact, there were 2 separate patches along the towpath, and we have no information from the net about those patches. The second issue relates to avoidance. So what you collect along the towpath or with a cast from a net, it seldom reflects what was present along its towpath. And this is because nets and the bridle and the tow cable all create hydrodynamic disturbances in the water that can be detected by pressure sensors on zooplankton. Consequently, a proportion of the animals in the front of the net are going to sense these disturbances and get out of the way. And that problem is more acute for larger zooplankton and micronecton, which are better swimmers. And that's why we typically employ larger, faster nets to collect larger, faster organisms. The longer a net is towed, the more problematic the avoidance becomes. And this is because the net becomes clogged with organisms and other debris, and this increases the pressure wave of water that's pushed ahead of the net. For animals with good vision, there's another challenge. The net may be visible to the organisms at a range sufficient to allow them to avoid it. And that's why, to partially overcome this, many nets, such as this 1 meter mock nest, are dyed dark colors to reduce their contrast with the background. The contrast is only part of the challenge. Bioluminescence stimulated by the struggling animals in the net can cause the net to light up, which stimulates further avoidance. Now, there have been attempts to avoid this, to overcome this. Peter Wiebe at the Woods Hole Oceanographic Institution, or WHOI, has demonstrated that a mock nest equipped with a forward-facing strobe light collected significantly more euphausids than a non-strobing net, presumably because the strobe startled and blinded the euphausids, preventing a directed escape response. The third issue created by nets is that they're really hard on fragile gelatinous forms of zooplankton. Since gelatinous zooplankton may predominate the community at certain times and in certain places, our understanding of pelagic ecosystems was profoundly biased by our reliance on nets. Rather than collecting intact animals, gelatinous organisms are typically extruded or mangled inside the turbulent and often violent conditions inside the net. Well, collecting zooplankton is really just the first step in the process of turning samples into useful information. It's arguably the easiest and the cheapest part of the process because once the samples have been preserved, they require curation to maintain proper storage conditions. And then in order to process them, we require a lot of human expertise and time. So this takes time, and skilled personnel cost money. And as a result, many samples are collected, archived, and never really processed, or only partial contents are examined. And I mentioned this aspect in relation to nets, but I'm going to return to it later when we start talking about imaging systems, because there's a parallel issue that I think you can already begin to imagine. Well, you didn't come to this class to listen to me talk about nets, so let's shift our focus towards plankton imaging systems. Let's look at a hypothetical imaging system that might quantify plankton. In situ imaging systems take a series of pictures or images, or they collect a continuous image along the trajectory of the net. In this case, we have a camera and it images a discrete volume of water illuminated by a strobe. Each image is either stored internally or sent to the surface via cable where they're stored on a computer hard drive. So I've shown 5 images here, but if this example was from a system that was taking images 30 times a second or 30 hertz, this would be the result of just 0.167 seconds of data. So next, there are a series of steps that we need to take to process each image. First, we need to locate and isolate regions of interest within each image, and we'll call these ROEs. And we need to count the number of ROEs in each taxonomic category in each image. Now, we've only got copepods in this example, but we still need to decide a couple of things. How do we deal with partial organisms? Do we estimate the fraction of the whole? And that's going to be difficult if we don't know what how much of the total animal is that we're seeing. We could just count an animal if we see the majority of its prosome, and that's what I'm going to do here. So next, we divide the number of ROEs per image by the image volume, and we come up with a concentration. So let's assume that was a 10 milliliter image volume. We could estimate an equivalent concentration for a larger concentration. Say we could express it as numbers per liter. And then we'd average the concentrations from the individual images over a standard time or distance or a depth bin and calculate a mean abundance for each bin. Well, we wouldn't do that for just 5 images, but in this example, the system is taking images at, say, 30 hertz. It's just not practical for us to do this visually. And I know that it's not practical to process these images visually because I spent a good chunk of my life as a postdoc at Woods Hole scrolling through about 4.5 hours of video plankton recorder data, frame by frame. So it can be done, but it's not efficient, and you got to wait a long time for the results. So the camera and its vehicle are just part of the imaging system. The other part is the software that processes the images, and this is a critical part of it. And this system has to do several things. It has to store the images and their metadata, which would include location, depth, date, time, hydrographic information, and other measured properties. And then it has to isolate the ROEs from the background. This is a process we call segmentation. And as part of this, there may be a need to perform some focus detection, and only ROEs within some size range may be of interest. Then the users have to construct a training library that's made up of ROEs of the same taxa. And then we have to extract features that can be used to discriminate amongst taxonomic categories from each ROE. And these features can be things like length, width, area, bright grayscale, granularity. There's a whole lot of properties of images and of organisms that could be used to distinguish one from another. Just think about when you look at a copepod versus a chaetognath, you're evaluating features that you have that tell you what one is and what the other is. And we're trying to give the computer the ability to get as much information as possible to distinguish one image from another. Next, we have to build a classification algorithm, and that training, that has to be selected and tested and refined until its predictive capability meets our expectations. And then finally, the classifier needs to be run on unlabeled data, and errors have to be corrected. So without having computers to do this heavy lifting, we're going to collect hard drives full of images, just as we have repositories filled with jars of plankton, but we won't have data that we can use. So there are a lot of imaging systems out there, and many of them are one-of-a-kind or small production research systems. In 2003, Peter Wiebe and I published a review that covered most of the systems used to sample zooplankton. Understandably, it focused on net systems because they predominated, but we covered optical systems and acoustics as well. And even going back 20 years, there were a lot of optical systems out there. And you can see from this plot, which shows where different imaging systems appeared in relation to time, we need to go back to about the early 1950s to see where these systems first appeared. So people have been taking pictures of objects underwater for a long time, but the first attempts to image zooplankton in situ appear to have taken place in the waters off Japan. A scientist named Nishizawa and his colleagues published a paper in 1954 based on still images of live zooplankton that were taken from within a diving chamber using a collection box of clear plastic. And so the collection box was placed in front of and outside of this undersea diving chamber, and the box could be opened and closed to collect a sample of water and plankton. Nishizawa and his colleagues projected a collimated beam of light into the chamber from a 300-watt mercury vapor lamp, and this was pulsing at 50 hertz with a pulse length of 0.01 seconds. And then they took pictures with a Focus-Bell camera, and they were able to image a variety of particles in plankton, including copepods and chaetognath, at depths ranging from 10 to 75 meters. So here are a couple of images from that study. What's interesting is that they used a collimated light source, and we'll see this type of light source used quite commonly today. Its collimation wasn't particularly as good as the ones we have today, but it was still somewhat directed. They strobed their light at a fairly short duration, about 1/100th of a second, but the camera exposure time was much longer. And so you can still see there's some motion blur in these images, and that's because some zooplankton can move very quickly. And today, if you want to freeze the motion of zooplankton and eliminate relative motion from the moving camera system, you've got to pulse your strobe at only a few microseconds, and 1 microsecond is 1/1,000,000th of a second. And third, they were using dark field imaging, which means that you're relying on light reflected back from an object back to the camera. This requires quite a bit of light, and it's going to illuminate objects that are within the focal plane of the camera, as well as those that are too distant or too close to be in focus. So Harold Doc Edgerton was a pioneer in the imaging world, and he's most well known for his use of strobes to freeze motion. In the mid-1950s, there was a lot of interest in sonar returns from the deep scattering layer. And this was the impetus for Edgerton and his colleagues to design an in-situ camera system to ground truth the scattering layer. And so they developed a 35mm repeating flash shutterless deep sea camera in a pressure housing. You can see the camera is aimed forward, and there's a light projected more or less parallel, but it's going to diverge, and the camera is going to image target somewhere out here, somewhere out in this area. And they were successful in collecting images of larger zooplankton such as salps and micronecton that appeared to be associated with these strong acoustic returns from the deep scattering layer. And this is something we see repeatedly today, and that's the use of acoustics and imaging together. So acoustics being used to ground truth scattering layers, or acoustics being used to provide some context so that we direct our imaging systems into water where there may be something of interest. This next example came from a freshwater application in Germany. Schroeder was a graduate student, and he built this system using a Grundig TV camera, which he borrowed from the manufacturer. And then he aimed the camera into a volume of water that was illuminated by a pair of lamps oriented at 90 degrees to the camera. And this approach is useful because he was focusing only on a region where the water was illuminated. So it helps to eliminate most out of focus organisms. And since the light that is scattered only light that's scattered at 90 degrees to the camera will be returned to the camera, you need a pretty strong light source. So he had 2 lamps. This has a kind of a sad backstory. Schroeder borrowed the camera. He was a graduate student, and after he was using it for a while, the whole system flooded and the camera was destroyed. So I'm sure there were some repercussions, but nonetheless, really interesting study. And this kind of setup isn't that different from some of the systems we'll see that are much more recent. This development isn't an in situ technique. In fact, it's an approach that was designed to facilitate the processing of preserved plankton samples from nets or pumps. Peter Ortner developed a method where you took a plankton sample and poured it onto a piece of photographic paper in a dark room and then briefly exposed it to light. This created a silhouette of the contents of the sample that could then be processed under a low-powered microscope, sometimes with a grid overlaid to allow subsampling. And I mentioned this approach because it was a precursor to the use of flatbed scanners and instruments such as the Zeus scan. As I mentioned earlier, nets have problems documenting fine-scale patchiness, and they're particularly hard on fragile gelatinous organisms. They also don't do well in murky water, in turbid water. And so one attempt to overcome this was to mount a camera near the cod end of a net to image organisms as they passed into the cod end. And as you can see, this is an effective way of documenting both fragile taxa, and it also provides a record of some of the finer scale patchiness. This system was capable of imaging at 1 meter intervals. And it also allows work in more turbid water because the path length between the strobe and the camera is greatly reduced. It's constrained by the diameter of the net just in front of the cod end. This system did work quite well, but it was limited by the amount of film that could be stored. It was a 35mm mechanical camera system. It did, however, reduce sample processing time, and a paper 1993, only in Ed Hood estimated that there was a mean saving of about 24.6 hours per sample by using this camera net system. So an evolution of the camera net system was the incorporation of a video camera into the cod end instead of a still camera. And the ichthyoplankton recorder employed a video camera mounted in the front end of a high-speed Gulf 5 plankton net. And separate interlaced video fields were telemetered to the surface via a conducting cable, and they were recorded on VHS tape, SVHS tape. It imaged a volume of about 4.5 milliliters, and the primary advantage of the ichthyoplankton recorder over a 35 millimeter camera system were the increased horizontal spatial resolution and an increase in tow endurance. So it's not just battery powered now, it's getting power from the surface and it's sending the data back up to the surface. The video and the environmental data were telemetered to the ship while the system was towed at 5 knots. And so this sampling rate and tow speed gave them an estimated horizontal spatial resolution of 3 to 5 centimeters. Data from the IR system, the ichthyoplankton recorder, could be continuously recorded on SVHS tape with only brief gaps when they were changing the tape. So what we see here is a pretty big advantage. Video data being sent directly from the instrument to the ship where it was stored on videotape. Now at the time, the video resolution was quite low, and so this limited the area and hence the volume of water that could be imaged. So next we have the video plankton recorder. And this was developed by Cabell Davis and Scott Gallagher. Scott's here in the room at Hui. And this is a picture of one of the early versions of the VPR. And the VPR telemetered the data to the surface like the ichthyoplankton recorder, but it did it over an electro-optical cable. The original VPR that you see here had 4 video cameras, and each camera imaged a concentrically located volume of water ranging from less than a milliliter to about 1,000 milliliters. The image volumes were illuminated by a high-intensity, short pulse, about 1 microsecond xenon strobe that was synchronized to the sampling rate of the camera. So when the camera shutter opened, the strobe pulsed. And the strobe was about 1 meter from the cameras and oriented to provide dark field illumination. The cameras and the strobe were mounted on kind of a prong-shaped vehicle, so they're imaging undisturbed water. And so this was designed to reduce avoidance. And the whole system is equipped with a CTD and other hydrographic sensors. So the VPR was capable of producing some very clear images of zooplankton while being towed at a fairly high speed. One of the challenges posed by imaging an unconstrained volume of water that's illuminated by an obliquely mounted strobe is to determine the depth of field. And although the width and the height of the image were clearly defined by the limits of the video field, the depth was more problematic. Each video field can potentially contain in-focus images of organisms, as well as organisms located either in front or behind the image volume. So here you can see the in-focus region in that little dashed line, and so that'll produce an image somewhat like the one you see. But then we can also have organisms that are located outside of that field of view, and that's going to produce an out-of-focus image. Sometimes the out-of-focus images are still recognizable, and that creates a challenge. Do you count those animals or not? So we have 2 copepods in this image. One's in the focal plane of the camera and the other one isn't. One's identifiable as a copepod. The second blurry one might be a copepod, but we don't want to count the blurry one. So you have to train, if you're doing this visually, you have to train the observer to be pretty consistent in how you count animals. You'll also notice that the out-of-focus copepod is further away from the camera and it appears smaller. And that's because this system isn't telecentric. And telecentric means that objects stay the same apparent size regardless of their distance from the camera. But if it's not telecentric, things will get apparently bigger as they get closer to the camera. So there are a number of different variants of the original VPR. It used an analog standard definition video camera. You can see the one in the top left has just 2 cameras on it. Here's the one on the top right is on a tow body with a pair of cameras. The one on the left is a vertically profiling system, an autonomous system that Scott developed, and this would be deployed in a trawl-resistant shield on the bottom. It would periodically float upwards, collecting data, and then be winched back down. And on the right, you can see a surface skimming system attached to a boat. There have also been flow-through systems. The VPR was used very effectively as part of the U.S. Globex Georgia Spank program in the Northwest Atlantic. And it was put on a vehicle called Biomapper 2. And Biomapper 2 incorporated a single camera analog DPR on this vehicle, Biomapper, which had 5 pairs of upwards and downward facing acoustics, as well as quite a complex hydrographic sensing system. And so the video data were sent to the surface in near real time, where an image processing system performed segmentation and focus detection and extracted ROEs in real time and wrote them to a hard drive, while the raw video was archived on videotape. And so this is, if you were viewing the image processing system at sea, you would be seeing these ROEs flashing across the screen. They were written to the hard drive. Unfortunately, the image volume was quite small due to the relatively low resolution of standard definition video. So to achieve adequate resolution, you couldn't look at too large a volume. And this meant that we often see only parts of large animals. So it's difficult to identify things like siphonophores, scalp chains, medusae. If all you see are some tentacles, it makes the identifications quite challenging. And we know that, you know, this kind of problem is hard. Depending on what part of an animal you see, you're going to come up with a different hypothesis as to what it is. So there were a lot of unidentified things, particularly these larger organisms. But a big advantage of Biomapper 2 was the combination of an imaging system with acoustics, multi-frequency acoustics. And using an imaging system with a small volume to survey large quantities of water doesn't really make much sense. There's a lot of empty ocean out there. But we know that animals tend to be concentrated along hydrographic features such as chlorophyll maxima and thermoclines. And acoustics can see the echoes from these organisms, and then the camera can be guided into those features to determine the composition. And so here's an example of this use of acoustics and imaging from the Gulf of Maine. This is from a paper that Andoni Lavery, myself, and others published. And you can see here we've got 5 frequencies of acoustics, 43, 120, 200, and 420 kilohertz. And the bright red echoes are stronger echoes and the blues are weaker echoes. The towpath of the Biomapper 2 vehicle is seen along here. And the circles, the white circles, represent the presence of larval physonect siphonophores called siphonula. And each one of these little siphonulae contains a carbon monoxide bubble. And that carbon monoxide bubble, which you can see in these ROEs here, it rings, it resonates at certain acoustic frequencies, rings like a bell, and gives us this strong scattering. So these patches didn't necessarily reflect dense concentrations of organisms. They reflected these larval siphonophores that were very strong scatterers. So at about the same time, over in France, a system called the UVP was being developed. Now, the VPR was primarily a towed instrument, but Gaby Gorski and his colleagues at Villefranche-sur-Mer, they developed a video-based profiling instrument called the UVP. It was originally designed to image marine snow, not zooplankton, but it proved quite effective at quantifying larger zooplankton as well. And so basically it consisted of a video camera that was oriented down, and it was aimed into a collimated light sheet coupled to a CTD, a data logger, and batteries. The light sheet was about 90 centimeters from the camera, and it could be produced either continuously or as strobe light. And the continuous illumination gave you stronger light, gave you a parallel beam of about 150 watts. And it illuminated a fairly large field of about 20 centimeters by 14 centimeters by 1.5 centimeters thick. And so it was looking at a larger volume, so this gave it lower resolution than the VPR, but it was better suited to imaging larger objects because you saw more of them. So here you can see just a schematic of the UVP3. and some example images. So you can see larger medusa, some acantharians, some decapod, or some shrimp, and it looks like some ctenophores. This coincided with the time where we were starting to see a lot of new sensors develop. So consumer digital cameras were emerging, and they had either CCD or CMOS sensors, and this allowed the incorporation of higher resolution digital sensors into imaging systems. And we also start to see development of image processing software. You saw it with the VPR, and the VPR had a MATLAB-based image processing suite that could perform all of those tasks that I described earlier, starting with segmentation, going through to classification. Just as a digression, in 1998, the VPR and the UVP were expensive systems. Peter Tiselius in Sweden developed a low-cost variant of based on the VPR. He called it the RIG, and it was given the name the in situ video camera. It used PVC housings and a lower cost video and strobe system. Right, so moving away from in situ systems for a moment, we have the flow cam. So in 1998, Chris Saraki and colleagues at the Bigelow Laboratory published a paper in MEPS describing a benchtop system for rapid counting, imaging, and measuring of individual cells and particles in the 20 to 200 micron range. This system was commercialized by Fluid Imaging Technologies, and it's evolved into a series of instruments that include color cameras, higher resolution cameras, and larger volume flow cells that are capable of imaging mesozooplankton. And it uses a software suite called Visual Spreadsheet to process and classify the particles. And you can see how the FlowCam has evolved from those early images. These are some example ROEs of phytoplankton and phytoplankton and some microzooplankton imaged by the FlowCam. The quality is very high. The company was purchased by a Japanese company a couple of years ago, Yokogawa, and it now markets and develops the FlowCam system. And so I'm just going to play you a brief video from Yokogawa Fluid Imaging to show you some of the operation of the current system.
This is where the FlowCam comes in. FlowCam's revolutionary technology combines the benefits of digital imaging, flow cytometry, and manual microscopy into a single instrument. Here's how it works. After a liquid sample is introduced into the top of the FlowCam, a software-controlled syringe pump pulls it through the flow cell and past the FlowCam's optical system. Using a stroboscopic flash to eliminate motion blur, a camera captures images of algae and other particles in the sample through an objective lens. Simultaneously, an optional onboard laser excites fluorescing pigments present in specific organisms, enabling the FlowCam to automatically distinguish between groups such as cyanobacteria, diatoms, and other types of algae. In real time, the FlowCam software, Visual Spreadsheet, automatically separates and catalogs the images of each organism. Using built-in image recognition tools, users can classify organisms, estimate biovolume, count cells, and calculate the abundance of each taxonomic group. Drinking water utilities use the FlowCam to proactively monitor water quality and safety, detect the presence of cyanobacteria, and stay ahead of taste and odor issues. Biological oceanographers study primary production and track the presence of toxin-producing organisms to better understand and monitor harmful algal blooms. Labs all around the world are using the FlowCam to streamline the process of algal analysis. If maintaining healthy water systems is important to your organization, download our ebook to learn more. FlowCam, a trusted partner worldwide since 1999.
So Harry Nelson is in the room here, and I know Harry's going to talk to you in a subsequent class. So he probably knows more about the flow cam than anyone on the planet. So you'll learn a lot more when he
Is where the flow cam.
Returning to systems in the water, in the late 1990s, we also saw the development of the first line scan system. So line scan cameras don't image an area, they image just pixels along a line, but they do this very rapidly. And so all of the images I've described so far take discrete images separated in space. And so this means some organisms will be missed and others will only be partially imaged. So this completely new type of system was developed at the University of South Florida called SIPR, the Shadowed Image Particle Profiling and Evaluation Recorder. And this camera produced a continuous swath image of water as it flowed unidirectionally through an intake. You can see the intake just up here, that black area in the center of this larger vehicle. And it flows through this intake and it passes through a 96 millimeter by 96 millimeter tall, 1 millimeter thick continuous light sheet that's formed by a laser. And light from that sheet is then focused onto a digital line scan camera. SIPR contains a pair of light sheets and camera systems allowing images to be collected from two planes rotated by 90 degrees. One system employs a line scan camera with a 4096 pixel array. The other uses a 2048 pixel array. And these cameras provide a continuous digital record of targets that pass through the light sheets. The data are stored on high-capacity hard drives, and images of the targets are reconstructed using a custom image processing software. The system can provide a continuous record of the contents of up to 30 cubic meters per hour. So we're starting to see a much larger volume than typical imaging systems. And here are some examples of images from SIPR. On the left, you see just part of a raw image. It's capable of producing some very clear images of zooplankton, including larger organisms. It's a black and white system, and in some ways, it's an in situ version of Peter Ortner's silhouette photography approach. The system uses custom software to perform the segmentation and extraction of organisms. It also uses a semi-automated identification algorithm, and a flow meter corrects for distortions in the images induced by changes in velocity. So in the 2000s, the advent of consumer and industrial digital cameras really provided an expansion in imaging systems with a huge advance in the form of greater resolution. And this allowed image areas to increase while still maintaining acceptable resolution. The higher bandwidth required for these systems necessitated the use of fiber optic cables. Image processing systems on board improved the segmentation and focus detection. And you can see here from digital video plankton recorders how dramatically better the quality of these images are. You really start to see a lot of detail. You also have greater dynamic range in some of these cameras, so you have more grayscale values that can give you clearer images. So in the 2000s, we saw rapid improvements in digital camera technology, and in parallel, we started to see a proliferation of digital plankton imaging systems. Higher resolution cameras were added to the VPR. New optical setups provided better defined and larger fields of view. And the addition of color cameras provided new taxonomic features that in what had previously been a grayscale world. So here we can see Dougal Lindsay and his student Jessica Pretty with a color VPR doing a cruise to the Chukchi Sea. And these are some of the ROEs from the color VPR. So you have a whole new set of features that you can use, plus just a new gestalt for the world of plankton. It's not a black and white grayscale world. It's a very colorful world. So the digital VPR was built into a fast active undulation system called the Flying Fish. And this allowed the vehicle to fly like an underwater airplane. So it could cover much more of the water column because it was capable of much steeper angled toyos. And this was combined with a sophisticated image processing system that incorporated focus detection, size selective thresholding, feature extraction, and an automated classification system. And as a result, it was finally possible to obtain near real-time data on zooplankton distributions while at sea. So here you can see a plot of that's color-coded of the number of Calanus finmarchicus per liter along these very steep toyos from the flying fish. So let's take a look at a short video where Dennis McGillicuddy at Hui explains the operation of the flying fish during a cruise. I think they were looking at Trichodesmium.
In April 2018, research vessel Neil Armstrong set sail from Woods Hole to investigate changing plankton productivity. One of the key instruments the scientists use to measure the abundance and distribution of plankton in real time is the video plankton recorder. Dr. Dennis McGillicuddy and his team adaptively sampled the ocean with this towed underwater video microscope system, right here at the shelf break front.
We spent the last 14 days looking at the productivity of the shelf break front region, which is the outer edge of the continental shelf here. There's a strong front that separates the relatively cold and fresh waters of the continental shelf and the warm and salty waters of the continental slope. We've been focusing on this line of stations here, which is couched within the Ocean Observatory's Pioneer Array, looking at the productivity along the shelf break front. Partway through our expedition, we experienced a very unexpected bloom of a colonial form of phytoplankton called Phaeocystis, which appeared in the middle of our stations about halfway through the voyage. We were not expecting to find this bloom of Phaeocystis here at mid-shelf. It was quite a surprising finding, and we were very fortunate the very next day to get a satellite image which showed that, in fact, what we were experiencing were tendrils of this phytoplankton bloom coming down the shelf. So this is a satellite ocean color image where the redder colors are more enhanced in chlorophyll, the bluer colors are less so. And you can see these tendrils of this regional scale bloom coming down the coast. Now, we know we saw Phaeocystis here. We couldn't be sure that it was also Phaeocystis in this area. So we took the opportunity to use the video plankton recorder to do adaptive sampling, drew a course track that connected our sampling transect into the core of this regional scale bloom to see what we could find. Immediately upon putting the VPR in the water, we could see the colonial forms of Phaeocystis popping up on the screen like popcorn popping out of a movie theater popcorn machine. Just extraordinary abundance. So here's a plot of the ship's track as you see us going along east, and then north. What you're looking at is optical backscatter, which is actually a proxy for the amount of colonies in the water. I'm going to rotate it around so you can see the distribution a little bit better. But as we steam to the east, this area of high abundance near the bottom was extended higher and higher up into the water column. In the core of the bloom, the Phaeocystis colonies were present in extraordinarily large abundances from the surface all the way down to the bottom. We also noticed as we turned the corner and looked at the forms of the colonies, there was a dramatic change. Some were transparent. As we approached the inner core, we were seeing a lot of more opaque colonies, and we think that the different kinds of colonies reflect different phases of the Phaeocystis life cycle. Having this information in real time allowed us to make water column measurements, collect water samples that could be used for microscopic analysis, DNA analysis to know exactly what species we're dealing with, full suite of nutrient measurements to understand the growing conditions for this organism and why it's so favorable for the production of Phaeocystis in this core region, and then the dynamics of that regional scale bloom as it's swept along the continental shelf. A bloom of this magnitude and size will clearly have important ramifications for the ecosystem. Phaeocystis itself actually is a form of phytoplankton that's generally less available for grazing by zooplankton and other creatures. So we expect it will have some important ecosystem ramifications, and now we have the data to go investigate that.
So the VPR continues to be widely utilized throughout the world, and it's a commercially available system from C-SCAN. This is definitely a design that has and will continue to evolve and see use for the foreseeable future. Large gelatinous plankton have been a challenge for imaging systems given the relatively small sample volumes available in most instruments. To overcome this, Larry Maiden and his colleagues at WHOI developed a light sheet system with high-resolution digital cameras aimed into the light sheet. You can think of them as the UVP turned on its side with a larger light sheet and a better camera. Topsy had a 2 square meter light sheet and recorded internally, while Lapis had a 4 square meter light sheet and sent data to the surface over fiber optics. The system on the left is Lapis, and the south chain is an image from that system in the Antarctic. While these systems weren't used extensively, their principle of operation reappeared a few years ago as part of WHOI's deep sea mesopelagic exploration system, and we'll take a look at that towards the end of this lecture. ZooViz was a system designed in my lab in the early 2000s. We used it on a series of cruises in a fjord in British Columbia, Canada, to ground truth acoustic scattering patterns in the vicinity of a sill near the mouth of the fjord. This system produced pretty clear images of euphausids and other zooplankton within an image volume that could be controlled by changing the focal length of the lens and moving the camera vertically and the light sheet housing horizontally. So it projected a light sheet horizontally into an area that the camera was aimed down into. The focal length of the camera was set so that the organisms within the light sheet were in focus. We could vary the image volume from 300 to 3,500 milliliters, and the data was sent to the surface over fiber optic. So here's an image from an image that was 346 milliliters of water, and you can see a pair of Euphausia pacifica in it. They're about 1 centimeter long, and the Roes on the right just shows the detail that's present in that image. Obviously, as you look at a larger area, you lose resolution. So here's one from 3.4 liters, and you can see 3 of the Roes from that. They're still recognizable as euphausids. You can still get some measurements, but the trade-off was reduced detail. So we used this to investigate scattering patterns near the sill of this fjord. Deploying imaging systems as standalone survey instruments is challenging, given their relatively small volume compared to nets. And so that was the case with ZooViz. We didn't want to try and survey euphausids in this deep fjord with the imaging system alone, but we used high frequency acoustics to give us some context, and we guided the system into the scattering features. And you can see here the density of euphausids on the top panel goes up as we go through areas of intense scattering. In 2007, there was a new instrument that was developed at Woods Hole. So far, with the exception of the FlowCam, we've just looked at instruments designed to image mesozooplankton, but there was still an interest in using imaging systems to quantify smaller particles in situ. And the imaging flow cytobot was a design that combined a flow cytometer with imaging optics to document phytoplankton and microzooplankton. It was developed by Heidi Sosik and Rob Olson at WHOI. I know Heidi's in the room today, and she's going to be talking to you more about this type of system in another class. This system pumps water from the environment through a flow cell, where filtered particle-free water is used to direct the cells through the focal area of the camera. That also reduces biofouling. There's a pretty complex optical setup, but the end results are incredibly clear images of particles in the 10 to 100 micron range. The system was subsequently commercialized, and it's currently available from McLean Laboratories, and it's in use all over the world. It's been upgraded to image particles up to 150 microns. Here's a short video from McLean that summarizes the imaging flow cytobot.
The imaging flow cytobot. The imaging flow cytobot, or IFCB, is an automated submersible imaging flow cytometer. The instrument generates images of particles in flow taken from the aquatic environment. The IFCB is an in situ time series instrument and a proven technology purpose-built for the rigors of an aquatic deployment. The IFCB can be used for phytoplankton ecology research, detection and monitoring of harmful algal blooms, water quality characterization, aquaculture applications, and underway sampling.
The IFCB is a complete research platform, a submersible imaging flow cytometer with multiple triggering channels, optimized to acquire images of particles in the range of sub-10 to 150 microns. The IFCB is an ideal platform for phytoplankton monitoring and research. The IFCB is capable of maintaining a continuous sampling rate of 15 milliliters per hour, storing up to 30,000 images every hour, with a high resolution of approximately 3.4 pixels per micron. Post-processing workflows handle feature generation, as well as genus and species-level organism classification. Various operational modes include full remote control capabilities of the instrument's routines, such as sampling, cleaning, and calibration. Rated to depths up to 40 meters, the IFCB is designed for long-term continuous operation with enough local storage, fouling prevention, and cleaning reagents for deployments up to 6 months. Optimal long-term performance is maintained using onboard predefined standards. The IFCB is contained in a 25 by 100 centimeter aluminum housing, submersible to 40 meters. During sample acquisition, water is drawn into the syringe via the intake port and is injected into the flow cell for analysis. The flow system of the IFCB uses a filtered sheath fluid that contains a biocide to prevent biofouling and to insulate optical surfaces from the sample material. Sample fluid is injected into the sheath fluid and into the flow cell, where it will intersect with the laser. Particles passing through the laser will scatter light. The IFCB filters these light pulses to trigger sample image acquisition based on user-defined particle size and chlorophyll thresholds. When the collected light pulse meets the threshold conditions, a flash lamp illuminates the particle and the image is captured. Following each sample acquisition, the generated images become available for offload. Images are also archived on board. Images are then post-processed offline for identification, classification, and particle counts. The IFCB stores onboard biocides and cleaning solutions, allowing for long-term deployments. Self-cleaning can be scheduled or interactively triggered. A calibration standard is also carried on board to allow for periodic checks of the instrument's performance. From real-time HAB monitoring to water quality characterization, the IFCB represents the next generation in advanced in-situ instrumentation. Contact McLean at mcleanlabs.com for more details.
In 2008, another system, a line scan system, was developed. This is the in situ ichthyoplankton imaging system, or ISIS. ISIS, despite its rather unfortunate acronym, represents a substantial advance in zooplankton imaging. Like SIPR, this is a line scan system. However, the imaging area is open rather than contained within the vehicle. This is the first time that we see an operational use of shadowgraph imaging, which provides a very large depth of field. Because the system is designed to be towed rapidly at 2.5 meters per second, it images a remarkable 70 liters of water per second while providing 68 micron resolution. Shadowgraph systems are also telecentric, which means the apparent size of the target does not change with distance from the camera. Shadowgraph systems are also capable of recording very subtle differences in density. You've probably seen shadowgraph imaging of shock waves behind aircraft and bullets. This same principle allows ISIS to image the internal structures of some gelatinous organisms. So here we can see some examples of ROEs from ISIS. As the name implies, its target is towards ichthyoplankton, and if you're going to look at ichthyoplankton, you need to look at a much larger volume of water. ISIS evolved from its prototype system with a single camera to a commercially available instrument called ISIS-2 that can be mounted on an active tow body. It has 2 line scan cameras, a large camera imaging 140 liters per second with 68 micron resolution, and a smaller camera imaging 15 liters per second at 59 micron pixel resolution. Each camera is producing a continuous picture that's then parsed into equivalent frames at rates of about 17 and 61 frames per second, respectively. Combined, the two cameras produce 660 gigabytes of uncompressed imagery each hour. So ISIS generates enormous quantities of data, and this requires an image processing system to support this data deluge. Fortunately, the system comes with software to extract ROIs, and a new paper describing their content-aware segmentation algorithm will be published shortly in Frontiers in Marine Science. So I'll just play a short video showing some of the ISIS imagery. Now, this is raw imagery. It's not yet corrected for variations in the background illumination. A very impressive system, and there are quite a few of these ISIS systems in operation. In 2010, the UVP underwent an upgrade, and a new instrument, the Underwater Vision Profiler UVP5, was developed. But although it was derived from the UVP3, it had several important changes. It was also developed by Gabby Gorski's team in Villefranche-sur-Mer. It has a camera aimed down into a horizontal light sheet, but it's internally powered and can be used by itself or in conjunction with a CTV. It has a 1.3 megapixel digital camera that produces images from a 22 by 18 centimeter field of view, and that light sheet is about 40 centimeters from the camera. It's produced by strobed red LEDs to reduce avoidance, and the camera is equipped with a bandpass filter so that only light from the LEDs comes in. This eliminates ambient light. It's capable of onboard processing of the images, and publicly available Zoo Process software is available to post-process the images. Its maximum deployment rate is about 3,000 meters. Here you can see the instrument near the surface and some example ROIs. UVP5 isn't designed to provide detailed images of small zooplankton. As is the case with all imaging systems, there's going to be a trade-off between image area and volume and camera resolution. As you can see from these example images, the UVP5 does provide fairly clear images of organisms of several millimeters or larger. The UVP5 was commercially available from Hydroptic in France, although it's recently been discontinued with the new UVP6 that we'll talk about in a moment. LOCI, or Light Frame On-Site Key Species Investigation, is an interesting system that was designed by Jan Schultz and colleagues in Germany to image plankton of 100 microns or smaller at high resolution while avoiding out-of-focus animals. It uses an optional net to pre-concentrate organisms, and it has a ring of 55 LEDs that produce a collimated light sheet around a Fresnel lens that illuminates particles from all sizes as they pass into the focal plane of a digital camera. So only organisms that are in focus are lit up. It uses this 1.4 megapixel monochrome camera imaging at 15 hertz and an optical setup that provides 13.4 micron pixel resolution. These images of diatoms illustrate the high resolution the system can produce for small particles, but it can also produce very clear images of larger particles. LOCI is still operational, and it's undergone developments, improvements to its camera, which is now a 4 megapixel camera, and it's been deployed in the Arctic and in oxygen minimum zones in the Southeast Pacific and elsewhere. Here's some more images from LOCI, and you can see that they are able to image larger plankton with it, presumably with a larger flow cell. In 2010, our lab began to develop a variant of ZooViz called ZooViz Deep. The Deepwater Horizon oil spill in 2010 created a need to quantify oil droplets and organisms at depths of over 1,500 meters. And so ZooViz Deep was a shadowgraph imaging system designed to internally record using battery power. Development of it took longer than it took to control the well, fortunately. And so it wasn't used during the spill, but it was used in post-spill monitoring cruises. During the oil spill, there was a shortage of research vessels, and many of the oil and gas supply ships were converted into research ships for science, but they lacked research winches with slip rings and conductive cables. And so we intentionally designed ZooViz to be self-contained and battery-powered. And I need to acknowledge the assistance of Rudy Strickler, who's a plankton imaging genius. And he really helped us to design a system that had an impressive shadowgraph imaging system. This allowed us to image about 140 milliliters per shot at a sampling rate of 2.1 liters per second. And with this system, we were able to collect some remarkable images of zooplankton and marine snow in the Gulf of Mexico. We also used an ISIS system for shallower sampling, covering a larger area. So the system has a 5 megapixel 12-bit camera. It's got a single board computer, two 750 gigabyte hard drives, strobe controller and power supplies. A lithium-ion battery gave it 8 hours duration. You set it up through either a Wi-Fi or a cable connection on deck, had an RBR CTD. The system could go to 6,000 meters, but the viewports were only rated to 2,000 meters. So here you can see the ISIS in the foreground and ZooViz, the first prototype ZooViz Deep being deployed in the Gulf of Mexico. And here's some example images. So this is a planktonic foram, 26% of the full image resolution. Some rows of various copepods. The scale bars are 1 millimeter. And just a collage of different organisms here at 48% of, well, this ctenophore is 48% of its full resolution. So a lot of detail and interesting structures, particularly of fragile gelatinous organisms. ZooViz Deep also did a good job of imaging small taxa, as you can see from these rowies, but it lacked an image processing system. We did later contract with Fluid Imaging Technology, and they were able to adapt their visual plankton software to help us process ZooViz images. Despite its long depth of field, there were still organisms imaged by ZooViz that were outside of the in-focus zone, but these images contained holographic information, and we were able to use this to refocus out-of-focus images with a MATLAB routine. So if we compare these out-of-focus images, just pick an image, and I'll change it to refocus. You can see how we're able to sharpen these images up by changing, by resampling the image using the holographic information. Now, imaging systems don't typically work very well in turbid water because of the absorption and scattering by particulate matter. Ortner's camera system got around this by reducing the path length so that the light had to penetrate a short path between the cod and the camera, between the strobe and the camera in the cod end of the net. An advantage that shadowgraph imaging systems have is that all of the light is directed towards the camera sensor, and a lot of it still reaches the camera under moderately turbid conditions. So in 2011, Hongsheng Bi at the University of Maryland acquired a ZooViz Deep system, and he deployed it initially in Chesapeake Bay. He's taken it to the Arctic and other locations. And it proved quite capable of documenting organisms ranging in size from ctenophores and medusae down to copepods in this turbid water. So here you can see on the right a full image with all these copepods clearly visible despite the very turbid water. We can see Heatignaths, a tautog fish, various gelatinous medusae and ctenophores. So Plankton Scope kind of evolved from ZooViz. It's an in-situ shadowgraph imaging system. It started out with the same basic design as ZooViz Deep. It was developed in China, and since then it's been refined, and it now includes the capability to survey plankton in coastal waters in applications ranging from moorings to towed or profiling instruments. You can see that it's now currently in the 3rd generation, and it's a commercially available instrument in China. Here are some examples of that Hongsheng B provided, showing data from the system being used near the entrance to a nuclear power plant, where Plankton Scope is providing monitoring and warning of blooms of animals that might clog the intake. So these mysids, you can see piles of them here removed from screens at the power plant, and you can see them appearing on ZooViz, and you can see the segmentation. He's got a scene-based classification system that does very effective segmentation and image processing. In 2014, we saw the development of the Scripps Plankton Camera System, or the SPC, and the SPC refers to a variety of cameras that were developed at the Scripps Institute of Oceanography by Jules Jaffe and his colleagues beginning in 2014. These systems have been deployed operationally with brief pauses since 2016. Advanced versions of these systems are still in use in other locations today as well. The SPCs produce a very large quantity of data, about 5 terabytes of foreground ROEs and about 2,000 terabytes of raw image data per year during continuous operation. It uses an image processing system that locates ROEs and saves them to a server array located topside. And the detail on these color images is really impressive. As you can see in these ROEs of Orthona, where we can see an individual without eggs, an individual with a pair of egg sacs, and another individual that has a parasite on it. There are at least 3 versions of the SPC: the micro SPC, which images organisms from 10 to 1,000 microns; the mini SPC, which images organisms from 100 to 5,000 microns; and the macro SPC, which images organisms 500 to 10,000 microns. Unfortunately, I wasn't able to get you any images from the macro SPC, as at the moment the SPC website is currently offline. However, there are a number of versions of the SPC in other locations. The Swiss Federal Institute of Aquatic Sciences, or EAWAG, operates a 2-camera system in a freshwater lake, the Griffin Sea, and they put out this short video showing its operation.
Now I'm counting and identifying the different organisms that are on the lake, and that's a time-consuming job.
So here we have the light sources, and here we have 2 cameras with magnification lenses, and a computer controlling the 2 cameras. The water is freely moving in and out in front of the magnification lenses, and the computer takes a frame every second during day and night, and we capture objects in the frames that correspond to plankton species. This website is where we publish in real time the images from the underwater camera in Greifensee. This is the low magnification camera images. And you can see, for example, the water flea Daphnia, or different type of copepods, different species. And one thing that we can already start noticing is that in many instances, we can image individual copepods that have other organisms growing on their body. This type of species interactions are generally lost when we sample with traditional methods for plankton. Accidentally, sometimes we also image some fish, and in this case, we have a beautiful parasite attached to the tail of this very small fish.
We are pretty happy that we can follow the dynamics of different species, a large number of them, which was impossible with previous automated monitoring systems. And we also see a number of organisms that we still don't know what they are.
So although the SPC website is down, you can go to the Aquascope SPC site and see those kinds of images, and they're updated daily in different size categories. The ZooCam is a shadowgraph telecentric imaging system that's mounted on a spray glider. The system is designed to autonomously image plankton as the glider moves through the water column. The design of this camera is similar to ZooViz and the plankton scope. Illumination is provided by a red LED that's collimated and reflected through a 45-degree mirror before passing through an optical viewport. It moves across 15 centimeters of undisturbed water, and then the collimated light returns to the camera housing, is reflected by another 45-degree mirror, and passes through a lens that focuses the image on a 1.2 megapixel camera. This takes images at 2 hertz with an exposure time of about 30 to 94 microseconds, and the image frames are saved at a rate of up to 2 hertz. This system was developed by Mark Oman at Scripps, and it's been used in a number of glider deployments. The image volume is 250 milliliters per frame, and the system acquires non-overlapping images at approximately 5 centimeter vertical intervals while the glider is moving at 10 centimeters per second. So it produces some outstanding images, as you can see from these examples. On recovery, all the images are corrected for uneven background illumination using a flat fielding algorithm, and then they're segmented using a size selective algorithm, and the resultant ROEs are classified using a convolutional neural net classifier followed by manual validation, and just another collage of some of the images that ZooCam is capable of producing. LISA is a shipboard system that was developed by Phil Culverhouse as a vessel-mounted high-speed monochrome line scan system, continuously collected to water pumped onto the ship from a depth of about 4 meters as the ship is underway. It images all plankton greater than 100 microns in a flow cell at 12.5 liters per second. The system is linked to PIA, which is the plankton image analyzer that performs the segmentation and ROE extraction followed by automated classification. And this system has been updated into a commercially available instrument called PI, which is a plankton imager, and it's a shipboard flow-through color line scan system that images organisms in a flow cell at 22 liters per minute, and you can see a color image here, and you can see some examples of the LISA, or the plankton imaging system, which is sold by a company called Plankton Analytics. So very impressive, and again, you gain color in here, which gives you additional features for classification. In 2017, the University of Sao Paulo produced a shadowgraph imaging system. This was developed by Rubens Lopez, I think with assistance from Rudy Strickler. It uses a collimated infrared LED beam and a 4 megapixel camera. It's got a frame rate of 6 to 15 hertz, pixel size of 5.5 microns, and a volume of 4.5 microns per shot. And here you can see the images. Computer vision tools developed at the Laboratory of Plankton Systems are used for classification and characterization of the different taxonomic groups, and they use a random forest classifier. It captures particles larger than 5.5 microns, but only particles 100 microns or larger are reliably identified. And you can see some examples here and vertical distribution of different organisms. WHOI is currently employing a new vehicle to explore and study the mesopelagic. This is called Deep Sea, which in many ways traces its lineage back to the Biomapper 2 vehicle that I showed you with a VPR and acoustics on it. This system has 3 imaging systems on board that trace their lineages back to Lapis and to holographic systems, which I just didn't have time to cover in this class. On I'll show you a short video that just shows the layout of the vehicle.
On board is one of the world's most advanced tools for ocean exploration. It's specially built to reveal the ocean twilight zone's mysteries. It's known as Deep Sea. It's equipped to visualize life from the smallest zooplankton to the largest whale in real time. It uses an array of sophisticated sonar systems used together for the first time, and an array of specially built camera systems for seeing animals nearby. A 3D camera for viewing mostly transparent animals. A powerful low-light camera to see animals actively avoiding the vehicle or not in its direct path. And a remarkable new holographic camera to see microscopic life in action. All combined, Deep Sea's multitude of cutting-edge sensors will allow scientists to get the most complete picture of life in the ocean twilight zone to date.
Another currently available system is the UVP6. This is a commercially available instrument from Hydroptic that's the latest evolution of the UVP series of imaging systems. There are two different units, the UVP6 LP or low power and the UVP6 HF or high frequency. Both of them are much smaller than the UVP5 and they differ in terms of power consumption and their data rates. The UVP6 has a 5 megapixel camera containing a motherboard with a processor, an image processing unit, an image sensor board, a lens, and a bandpass filter centered at a red wavelength, 635 nanometers. The light unit contains a controlling board, a laser diode, and lenses, and it has an onboard image processor that performs image illumination, equalization, segmentation, and size-dependent ROE extraction and storage. All the UVP6 instruments are intercalibrated so that data from one can be compared with data from another, and it uses EcoPart and EcoTaxa software for more advanced image processing and classification. And you can see that the UVP6 provides improved image quality relative to the UVP5. The blue bordered images are from the UVP6 LP, and the green bordered ones are from the HF. These systems are designed to be used on autonomous vehicles, on floats and gliders. You can see various applications where the UVP6 is on an Argos float, on a Sea Explorer glider, and on a mooring off Svalbard. So we've gone through a lot of different systems, and we've seen there have been incredible advances in imaging systems. The emphasis here is on imaging systems. In the beginning, we just had the hardware, and we relied on humans to do the heavy lifting of sorting through the images. And this is impractical, and it's really a parallel problem to the one posed by NETS. It's far easier to collect samples or images than it is to turn them into useful information. We've seen a lot of advances in the hardware, better cameras, higher frame rates, onboard image processing, greater dynamic range, shadowgraph imaging, line scan cameras, and color cameras. We've also, and these advances are really allowing us to look at larger volumes of water without sacrificing resolution. In terms of software, we've seen better segmentation algorithms, more diverse classifiers, and by that I mean capable of identifying larger numbers of different taxonomic groups. And we have classifiers that are capable of working well above 90% accuracy. We've also seen the advent of autonomous systems thanks to low power consumption and attempts to intercalibrate systems. All of these advances are helping us to reduce the time between data collection and visualization of useful data. And I think that we're going to see the cost of these systems come down as they become more widely adopted. So I think I'll stop there. Before I conclude, I want to apologize to anyone who may have a camera system out there that I didn't mention. I'm sure I missed quite a few. The innovation and ingenuity that are reflected in the different imaging systems in this community are remarkable. I also didn't have time to discuss holographic systems or benchtop systems such as the zoo scan. Back in 2003, when Peter Levy and I reviewed zooplankton sampling and sensing systems, imaging systems were a relatively small part of the story. Now they probably merit a new review devoted entirely to these technologies that are providing us with a remarkable picture of the plankton communities in our oceans. Given the advances that we've made in the past 70 years, it's exciting to think of where we're going to be in the next few decades. So thank you very much for your attention.
Thanks a lot, Mark, for the wonderful talk. It's a very comprehensive presentation of the evolution of plankton imaging systems. I would like to open the floor for questions, and those who would like to ask Mark any questions, please use the hands tool. We have Marco Ojales. Please, Marco, proceed.
Hello, Dr. Benfield. Thanks for the talk. It was very impressive. It was great to see the whole history behind the systems. It was interesting to notice that some of these systems are used because a lot of people like just develop these systems, and some of them are getting used more than others, or they become more prevalent through time. From your experience, what makes one of these imaging systems like become prevalent and be used through time and become then like a bit more of a standard to measure biological properties?
Well, we have people in the room who developed systems that were commercialized. So Scott Gallagher, Heidi Sosik, Harry Nelson can probably speak to this issue as well. I think part of it is publishing in good journals and getting information out, showing the utility of the systems. I think a second part is having the systems used as part of large oceanographic projects. So the U.S. GLOBEC program and the use of the VPR in that system, I think brought a lot of attention to it. I think those are two of the aspects, and I think many of us are not good in terms of being entrepreneurs. We're used to building these research instruments, but we don't think on the business side of things. I think a lot of our universities would like us to do more of that, to commercialize a lot of these kinds of technologies. So I think we have to change our mindset a little bit, but I think getting the information out there and using these systems and demonstrating their utility is a big part. I'm sure Heidi and Harry and Scott might have some ideas on that one too.
Before we proceed, I would like to ask for those who can open your cameras, because I would like to make a very nice screenshot of this beautiful audience. So please, thank you. In the meantime, Rainer, have a question?
Yeah, I might want to comment on Marco's question as well. And I think that also the availability of the processing software, or the quality of the processing software is also a decisive component of the success of an instrument or not success.
Absolutely. Yeah, that's a critical part.
And it won't proliferate.
Yeah, I think that's a really important point that you raised, that it has to be a complete system. If it's just a beautiful camera and it lacks the ability to turn those beautiful pictures into something meaningful and useful, then it's not going to be marketable.
Anyone else?
Harry, did you have a comment? I think you're muted. Still muted. Oh.
Yeah. There's something wrong with your microphone, Harry. No, not yet. Okay.
Maybe just put it in the chat.
Yeah.
Yeah, sure.
Chat.
Anyone else would like to ask Mark Benfield a question or a comment? Feel free to speak. Scott?
Hi, great talk, Mark. Very comprehensive. I think, to answer your question about, or address at least the question related to why do some systems persist in time and when others don't? And I think the reason that you touched on is persistence, and evolution. So things like the VPR are still being built and still being used, but we've gone ahead and evolved in such a way that we are now making a VPR-like instrument that is handheld and can be taken anywhere and put on a CPD rosette. And this is called CPIX, for continuous imaging and plankton classification system. And as a system, it is designed specifically to take the image and extract the ROE and then do real-time classification, or you can sync it with a deep learning classification system that allows you to organize images and do that on your desktop. So it's the evolution, I think, and that's been commercialized by Coastal Ocean Vision. So the evolution of systems that improve their usability, I think, is what allows different technologies to persist over time.
Yeah, good point. Excellent.
And Scott, may I add that in a few weeks, I had to check the schedule, but Klaus Miller from Germany will speak in the course. He will give a talk. And I'm aware that he has a CPIX running at his coastal observatory. And most likely he will talk about it, and you are more than invited, more than welcome to interact and also share with us your experience in developing the system and the applications around the world. I'm aware that you had the system running in Japan for quite some time as well. So we are very interested to learn more about it, about CPIX. And I would like to extend the invitation to Heidi Sosik, who I see here, because we will have also a talk by Lisa Campbell during the course, and she will talk about the flow cytobot. And I know that you are partners in this initiative in the Gulf of Mexico. So Heidi, I hope you will be able to be with us during Lisa Campbell's talk in a few weeks from now. I would like to ask Mark a question. Mark, about the cost of imaging systems. You said that as long as the commercially available instruments become more popular, their cost might decrease. But what about using new and cheap technologies to develop more affordable systems, especially for research institutions in countries located in developing countries? How do you see the situation in the present days?
So there are several examples of sort of crowdsourced open access systems. One of them is an instrument called planktoscope, not to be confused with plankton scope. But planktoscope does something similar to the FlowCam. It hasn't got all the bells and whistles of the FlowCam, but it's basically an imaging system to quantify phytoplankton and microzooplankton in color based around a Raspberry Pi. All of the plans for that instrument are available online. And I've purchased all the components on Amazon, and for about $500, we're going to build it this summer. You can have an instrument that produces remarkably clear images of phytoplankton. Our application is actually going to be microplastics, but so that system is out there and it will evolve, and it comes with the software to do the segmentation and classification as well. So that's an example. in situ instruments, there aren't as many examples of those, but the cost of industrial cameras, machine vision cameras, has dropped remarkably, and you can use pressure housings now that are made of plastic or acrylic that are lower cost. Raspberry Pis and things like that can provide lower cost. So I don't think it's going to be long, and maybe it's already happened, I'm just not aware of it, before there are going to be designs for in situ imaging systems that are based on off-the-shelf, low-cost components. I certainly hope so.
Okay. Rainer?
Yeah, hi, Mark. Based on your experience and review of all the systems around, what would you say are the most important aspects in designing plankton marine snow imaging system?
Well, having the software side to handle the deluge of images is going to be critical. So you've got to have software that can be used to process the images. I think stealth, making sure that you have something that is not going to disturb the water and reduce avoidance is really important. I really like the shadow graph imaging systems because they give you the ability to see animals that are nearly transparent because they can detect these subtle differences. Using red LEDs is another thing that I think is important. And then having something that can be adapted to different applications. Scott talked about CPICs, and you can swim with it, you can put it on a CTD, you can put it on a mooring. Having something that can do different jobs, because they are still expensive, and purchasing an instrument for tens of thousands of dollars or more, and having it only being able to do one thing is a challenge. And having an instrument that can be deployed from vessels of opportunity as opposed to very sophisticated research vessels is another advantage, because many parts of the world we don't have access to fiber optic slip rings and sophisticated instrument handling systems. So something that's lightweight that can be used. Those are a lot of things in that wish list, but those are some of the things that I think are important.
I find it very interesting that the DPR version was multi-volume, and I to some extent find it sad that we lost that idea to some extent, that we are only focusing on one volume and one size class, but sometimes also the volume that is imaged, is for a single image is too small, and you need to integrate over many images to get a good estimate of the plankton concentration, for example.
Well, there are, if we look at ISIS, ISIS 2 has two cameras in it, high mag and low mag. The SPC has, they on their mooring at the Scripps Pier, they have the two different magnification cameras. I think that there are still systems that can use two cameras, but you are creating a challenge. You're doubling your data rate potentially, or if you're not doubling it, it's increasing by some multiple. You're more complexity, more power consumption. So those are some of the costs. Now, holographic systems, which I didn't talk about, offer a lot of advantages because you can image a large volume, and within that large volume, you can still interrogate that volume and look at much smaller sections of it and see animals at high resolution.
Do you think we could get more holographic? Or?
Could you say that again? I'm sorry.
Do you think we should develop more holographic instruments or go more in the direction?
There are Scott probably knows more about this than I do, but there are holographic systems. I know there was one that Cabell Davis used during the Gulf oil spill, and there was a much smaller sort of one built in conjunction with MIT, which was sort of a handheld unit. So there are holographic systems, and as some of these telecentric systems contain holographic information as well that can be used to refocus organisms.
Yeah, by the way, we will have a talk about holographic systems on August 16th by Aditya Nayak, who's here in the room. So I think this will be a good opportunity to discuss further on the strategy, which is quite interesting and quite challenging as well, because the reconstruction process is not easy to implement, and it demands also a lot of effort. But it's a very interesting approach as well. Any other questions?
There was one from James Scott. He said the majority of the devices in the presentation were towed. He'd be interested to hear thoughts on the benefits, disadvantages of devices such as the plankton imager, which are onboard ship, limited to a fixed sampling depth compared to tow devices. So obviously, if you're taking in water from a ship at a fixed depth, you're only sampling one part of the water column, and we know that with vertical migration, you're going to see changes just day versus night from that fixed depth. I think they're useful for time series. If you're particularly interested in something that has a surface distribution, then maybe an egg survey, something like that, you can mow the lawn with the ship and cover a lot of ground. But if you're repeatedly steaming from, as the British Antarctic Survey does, going from England to Antarctica on regular cruises, putting one of these systems on there, it's almost like a CPR type survey where you're building up a time series, and you can start to look at impacts of global warming and other impacts on changes in the plankton community. So they have a different purpose. You wouldn't use them to get a full picture of the diversity of a plankton community based on just one depth, and of course avoidance of a big ship, which is going to create quite a pressure wave.
Different applications, different questions, Mark. Yeah. By the way, Nico wrote here in the chat about the plankton scope and curiosity, which is a low-cost stereo microscope, a kind of a stereo microscope, saying that they are good options for imaging in developing countries because of their low cost. I was reminding myself about the early days of scanning systems. By the time the ZooScan was developed, there were people using flatbed scanners, commercially available flatbed scanners, to generate images from preserved samples. And I recall you did that as well, Mark, if I'm not wrong. I know you have a ZooScan in your lab. But can you talk a little bit about that? Because I haven't seen many people recently using flatbed scanners to generate images, and perhaps that is related to the difficulties in standardizing the images from different instruments.
Yeah, so Phil Grosjean in Belgium was kind of the pioneer of using flatbed scanners and ZooImage software to be able to use an inexpensive scanner to quantify plankton. And Phil did it for a long time, but if you don't have someone kind of pushing that community forward, it kind of stagnates. And then we saw the ZooScan system. The challenge with ZooScan is that if you're using a ZooScan in Japan or the United States and you have problems with it or you need maintenance done, you've got to get someone, you've got to either ship it back to France or have a technician from Hydroptic come out. They may have representatives here who can do that now, but at the time when we were using our systems during the oil spill, when we had problems with it, it was challenging to get the technical support for those. But yeah, flatbed scanners and even silhouette photography still are easy ways, inexpensive ways of at least getting your plankton into some form of digital image that can be archived and that can be processed.
Okay, I have someone here Oh, Aidan Smith, would you like to make a question?
Hi there, greetings everybody. I'm Mark. Thanks for the very informative presentation. I just wanted to speak about the instruments and technologies that does the auto classification of plankton. What happens if the image taken only takes about 30% of the species, or maybe it takes a image of a species which is disorientated or on the sideways, on the backways? Is it still possible to classify those species even though it doesn't have a full image of the species?
Yeah, so that's an interesting question. What you're referring to is the fact that an animal can rotate in 3 dimensions, and a copepod viewed head on versus a copepod viewed laterally, dorsally or ventrally, might appear quite different. And so there's different solutions to that. You can have training libraries that have the different perspectives of an animal. So if you've got an animal that is going to appear quite different depending on the perspective relative to the camera, you can have a classifier that contains a library with lateral views and a library with head-on views. You can build libraries that rely on features that may be independent of the proportions of the animal. So there are features like grayscale values, granularity, roughness, texture, things like that, that might be a little more invariant depending on, regardless of what the orientation of the animal is. So there are a lot of ways to get around that. You know, when you have a zoo scanner or a flatbed scanner, you have the luxury of being able to orient the animals if something's in an odd perspective, but they're all lying more or less the same way. In the ocean, animals can be oriented in a number of ways. However, we've seen that many animals have a characteristic orientation in the water column. And so that means that in many cases, you're going to see the animal in more or less the same perspective if the camera system is the same perspective. For a glider, the zoo cam, they can correct for the angle of the vehicle to correct the image so it's normalized, so that you're not having rotation-induced differences in the animals.
Okay, noted. Thanks, Mark.
We'll have some talks later during the series of lectures dealing with
Scott had something to say about that. Yeah.
Dealing with neural networks. So from CNNs, we can extract even more features that are useful for exploring this problem of different orientations, for instance.
Yeah.
So I think this is going to be very interesting for us to learn about as well. Mark, speaking of that, I have a question related to the need to learn programming. So what do you think about the development of user-friendly software to allow people who are not so skilled in programming to analyze plankton images? Is this something possible in your opinion, apart from the examples you gave here with the flow cam?
Yeah, I do think it's important, and there are. I know that the folks who developed the ISIS are working towards a system that might be adaptable to processing data from other instruments, and so is the planktoscope software may have the potential. It's being used for imaging sonar systems and towed benthic camera systems. The same scene-dependent convolutional neural network deep learning classifier can be adapted to all those. Knowing programming is great, particularly if you're contributing to updating the software. If you've got some open source software and people are contributing new modules or advances to it, not having the programming background, as long as there's a mechanism to submit feedback to the developers so that new features and new capabilities and bugs can be sorted out, is fine. You know, I know MATLAB, and I don't want to learn another software package. So, you know, I would prefer, if I got some software as a classifier, I don't want to have to get under the hood and diagnose problems. I'd rather that it's pretty turnkey, and it's as simple as switching on the computer, launching the software, putting some parameters in, and throwing your instrument over the side.
Yeah. Well, I think this is one fascinating aspect of plankton imaging, because you are dealing with problems related to engineering, to the design of the systems, all the way from the biology, and then you also deal with the computer vision problems. There's a question, I think we have to finish with this one, by Tamar Inhay, asking about plans to combine imaging with DNA sampling.
Yeah, so Deep Sea is doing that in the mesopelagic. They're using eDNA, which are just fragments of genetic material in the water column that can provide you with an indication of the presence of an organism in that area, even if you didn't sample it. And so I believe they're using eDNA as part of their campaign with Deep Sea.
Excellent. And thanks, Mark Pichero, for pointing out about the EcoTaxa portal, which I invite all of you who doesn't know about it to access the portal, because this is a very, very important tool for all scientists and students working with plankton imaging.
Absolutely. The UV, I mentioned that with the UVP6, but that is software that could be developed and could be adapted to many different imaging systems as well. Thanks, Mark.
With that, I think we have to finish, because we're already about 2 hours online. So thank you very much again, Mark Benfield, for the wonderful presentation and for answering the questions and for your contribution to science in this field. And thank you all as well for being here, for sharing your interest. And I invite you to be here next week again for a talk about the FlowCam. We'll have Harry Nelson and Savannah Jude from Fluid Imaging talking about the instruments. So please come back in a week from now.
Thank you all. It was a pleasure to see a lot of familiar faces. So I really enjoyed it. Thanks.
Thank you. See you in a week from now.
Thanks, Rubens. Thanks, Mark.
Bye.
Thank you very much.